Lesson number 5, Feeding the World. By the end of this lesson, you should be able to evaluate the potential of biotechnology in relation to world food shortage. About 6.5 billion people live in the world. To stay alive and healthy, all of us need food, about 30 tons of it during a lifetime of 70 years. New technologies help farmers work the land so that food is produced in great quantities. For example, plant breeding programs produce new varieties of high-yielding crops. The new varieties are produced by crossing two different varieties, hybridization. Each variety has desirable characteristics, like resistance to disease and high yield, and crossing concentrates the desirable characteristics into the new hybrid variety, which is then reproduced asexually. For example, breeding new varieties of rice plants has boosted yields by more than 25% since the 1970s. Usually, a traditional plant breeding program takes up to 10 years to produce a new crop variety. Programs which use genetic engineering are much faster. This means that crops with desirable characteristics can be produced more quickly. As a result, more food, and better quality food, is available for people, particularly people in developing countries. For example, a new variety of rice, which makes vitamin A and is rich in iron, has been produced because of genetic engineering. Hundreds of millions of people could benefit from this development. Genetic engineering is one example of biotechnology we can use to produce more food. Another example is growing microorganisms for food in huge containers called fermenters. Single-cell protein, SCP, is a food produced from microorganisms. SCP could be useful as a foodstuff because it is fast-growing and has a high protein, vitamin, and mineral content. Microorganisms double their mass within hours, whereas the plants and animals we eat may take weeks and months to grow to full size. The microorganisms multiply using the nutrients in the fermenter as a source of energy and materials for their growth. Using the nutrients is an example of fermentation in action. Nutrient is replaced as it is used up. The fermenters are run for months at a time, during which microbial mass is removed at regular intervals and processed into food. SCP can be grown in large quantities in fermenters, which are a controlled environment independent of the weather and which take up little space. The technology is established and can be used in developing countries where food may be in short supply. However, do you think we might be reluctant to eat food made from microorganisms? How can we make SCP more acceptable? The issues which come from our ability to develop GM crops and produce SCP food stir up fierce emotions and vigorous debate. We can do the science, but should we do it? And if so, how best to do it?